Assalamu alaikum, dear students. Today we'll start a new activity in chapter 3, which is activity 8, specificity and mechanism of enzyme action. At the end of this video, you will be able to notice that the enzyme substrate complex is due to the complementarity of certain zones of the enzyme molecule and the substrate, relate the functional characteristics of an enzyme molecule to its spatial configuration and its protein nature. You need a pen and your notebook. In this chapter, we learned about proteins, how they are produced, their fate, etc. In this activity, we will learn about enzymes. Do you know what is the chemical nature of enzymes? Yes, enzymes are proteins. That's why we are interested in enzymes. Structure of an enzyme. Look here, miss. What can you see? Yes, we can see a long line of amino acids, that is how folded and coiled. So, enzymes are proteins. Their structure will be basically a long line of amino acids folded and coiled many times, showing a specific part called the active site. Look here. Here we can see a specific part of the enzyme, and it's called the active site of the enzyme. And we will talk later about its importance. Today we will learn about a property of enzymes, the specificity. What is meant by specificity of an enzyme? Does it mean each enzyme acts on a specific substrate? In fact, no. This is an incomplete answer. To discover the real meaning of specificity of the enzyme, you have to read the first point in this text. Enzymes have a double specificity. Specificity for the substrate, which depends on the complementarity of the spatial configuration or three-dimensional structure of the enzyme active site and the substrate in a lock-key relationship. So it's obvious that specificity of an enzyme has more than one meaning. First meaning, the specificity for the substrate, due to the complementarity of the spatial configuration of the enzymatic active site and the substrate in a lock and key relationship. So, the key has to fit in the lock. Look at the shape of the active site of the enzyme and that of the convenient or the specific substrate. How are they? Yes, they are complement. So the substrate has to fit in the active site as the key that fits in the lock. What about the second meaning of specificity of an enzyme? Let's read the second point in the text. Specificity in the reaction catalyzed. An enzyme acts in a specific chemical reaction on the substrate. For example, look here. We have an enzyme that catalyzes a reaction. One substrate is fixed in the active site of the enzyme. At the end, two products are got. This is a hydrolysis reaction. So here the enzyme is catalyzing a hydrolysis reaction. Here we have two substrates that combine in the active site of the enzyme to produce at the end just one product. So this is a synthesis reaction. Here the enzyme is catalyzing a specific chemical reaction, which is the synthesis reaction. So let's talk more about the hydrolysis reaction. In a hydrolysis reaction, one substrate is decomposed into two or more simple products at the end of the reaction. Here we have one substrate, and at the end we have two products. So in this case, the enzyme is catalyzing the hydrolysis reaction, or is acting in a hydrolysis reaction. In a synthesis reaction, two or more substrates combine together to give at the end one product. So in this case, the enzyme is acting in a synthesis reaction. In both cases, we have the formation of a complex enzyme substrate. Look here we have enzyme substrate complex, and all in all we have a hydrolysis reaction since we have one substrate and two products at the end. And here also we have an enzyme substrate complex, and this is the case of synthesis reaction since we have two substrates and one product at the end. So to sum up, we can say that specificity of an enzyme means each enzyme acts on a specific substrate, and also each enzyme catalyzes a specific type of chemical reaction. So these are the two meanings of specificity of an enzyme, and keep in mind that an enzyme substrate complex is formed during the chemical reaction. So here we have an enzyme substrate complex formed. Now, the active site of an enzyme. Remember when we talked about the active site of the enzyme? Where does the binding of the substrate and the catalysis of the chemical reaction occur? Yes, look here, in the active site of the enzyme. Okay, in your opinion, from what the active site of the enzyme is made up of? Remember, the enzyme is what? Yes, a protein. And from what a protein is made up of? Amino acids. So the active site is indeed made up of amino acids. So it's made up of amino acids since it's a part of the enzyme, knowing that the enzyme is a protein. Now, in order to discover the way the enzyme active site functions, you have to read the following paragraph. The enzyme active site, made up of amino acids, has two functions. First, to bind a specific substrate using binding amino acids, to catalyze the reaction underwent by the bound substrate using catalytic amino acids. So, amino acids of the active sites. Among these amino acids, I can talk about two groups. A group called binding amino acids binds to the substrate, and another group of amino acids called catalytic amino acids that catalyzes the reaction. Finally, to sum up, let's read in the summary. Each enzyme acts on a specific substrate and catalyzes a specific type of chemical reaction. 2. The substrate binds to the enzyme and the chemical reaction is catalyzed in the active site of the enzyme. The active site of the enzyme contains amino acids. A group of these amino acids binds to the substrate. Here I'm talking about the binding amino acids, and the other group catalyzes the occurring reaction, and here I'm talking about the catalytic amino acids. What about an application? Look here, miss. Here you have a figure that shows the occurrence of a chemical reaction catalyzed by an enzyme. You have to observe this figure and to answer the following questions. Take your time. Now, solution. Regarding the first question, indicate the type of the reaction shown. Now, the reaction starts with one substrate and it ends with two products, right? So this is a yes. Excellent hydrolysis reaction. Second question. Give the name of the complex formed in step 2. Here we have step 2. What do we call this complex? Yes, enzyme substrate complex. Third question. What can you say about the shapes of the substrate and that of the active site? So here you have the shape of the active site, and here you have the shape of the substrate. How are they? Yes, they are complement, right? And the last question. Give the name of the amino acids found in the active site that are responsible for the fixation of the substrate. Yes, they are the binding amino acids. Excellent work. Finally, don't forget to study the summary. Thank you for your attention. Goodbye.